Dr. Gaia Colasante is project leader at the San Raffaele Scientific Institute in Italy, where she leads a research team dedicated to understanding mechanisms underlying developmental and epileptic encephalopathies and in developing gene editing strategies to treat these disorders. She earned her degree in biotechnology from the University of Milan and her PhD in neuroscience and molecular medicine from the Vita Salute San Raffaele University. During her doctoral studies, she developed a strong interest in forebrain development, focusing in particular on the role of the transcription factors involved in cortical development. This led her to the Children's Hospital of Philadelphia, where she joined the laboratory of Dr. Jeffrey Golden, and there she continued investigating the function of those transcription factors in GABAergic interneuron migration. Afterward, she shifted to cell reprogramming. She identified a combination of transcription factors capable of converting fibroblasts and pluripotent stem cells into GABAergic interneurons, providing a potential source of cells for cell replacement therapies in epilepsy. Dr. Colasante is also leading in collaboration with Dr. Botani the first research project that we as the UGDH Foundation were able to award. And so we're very excited to hear from you now.
Thank you, Rebecca, for this introduction. I'm really excited to meet this community. So as Rebecca was anticipating, I've been studying developmental and epileptic encephalopathies over the last 10 years. And in particular, I'm interested in gene therapy for this disorder and in the development of mouse model that can help informing gene therapy studies. And I will show today something related to this, and then also something related to the project that the UGDH Foundation has funded. So I just wanted to start with this a definition of developmental and epileptic encephalopathies, which is, in my vision, relevant to understand why we want to treat this kind of disorder essentially with the gene therapy. So they have two main symptoms, as you all know. On one side, severe seizures, severe epilepsy, and on the other side, important neurodevelopmental alterations. And essentially, the cause underlying seizure and developmental impairment is essentially the gene mutation. There is also a component of the developmental impairment which is caused by seizure severity and by their impact on the brain. But anyway, the gene mutation is contributing directly to both of these phenotypes. And so you can pretty well understand that to really address both these phenotypes, it's not just sufficient to use a very good anti-seizure medication, because in this case, we will just address the component of developmental impairment that is a consequence of seizure. So we need to really remove the same cause of the disease, which is the gene mutation. And so I will bring today my experience in a very severe developmental and epileptic encephalopathy, which is Dravet syndrome. It is characterized by three different stages. A febrile stage that is the onset of the disease. So these babies start to have the first seizure during the first fever of their life, or during a hot day, or during the first bath. And there is then a worsening stage during the second year, in which the seizure worsen and also the first start to appear. And then a chronic stage in which the seizure start to ameliorate, also thanks to the therapies, but there is the sleep alteration, the cognitive impairment persist. And also these kids undergo a high incidence of sudden explained death in epilepsy, especially after a prolonged and especially severe seizures. So in this case, the genetic condition underlying this disorder is an heterozygous mutation in SCN1A gene, which is encoding for the alpha subunit of voltage-gated solute channel NAWA.1, which is a channel relevant for neuronal function. Indeed, this channel is essentially expressed in the population of neuronal population of GABAergic interneurons, which is the main source of inhibition in our brain circuit, especially in the hippocampus and the cerebral cortex, which are mainly related to epilepsy. And it is located in this part of the neurons, very specialized, which is the axonal initial segment. That is where action potential are generating. And so it's the point where neurons decide if they can excite and so generate an action potential or not. And so in a condition in which we have two copies, so two allele of the functioning allele of SN1A, we have the right amount on NAWA.1, and so the neurons can work properly. But in a condition of haploinsufficiency, so in which one of the two genes is mutated, we have half of the channel, functional channel available. And this causes an hypofunction of GABAergic interneurons that also alters excitation and inhibition balance into the brains with a consequent seizure and behavioral alterations, which are present in patients, as I was telling you, and also very well resembled in mice. And so there are two main strategies to address Dravet syndrome from a precision medicine perspective. So one is a gene supplementation strategy, and I was happy to hear yesterday that there is some research ongoing already for UGDH as well. And so the possibility to use a viral vector to deliver a healthy copy of the gene in order to replace the one which is not functioning anymore, and so to restore in this way the correct gene dosage. And the other possibility in this case that is actually not possible for UGDH is the presence of the healthy allele. One healthy allele is still there, and so we can kind of boost the expression of this allele in order to make it produce all the channel which is required to restore the proper function of neurons. However, although interneural dysfunction is the main initial, the main pathogenic mechanism of disease, actually across the disease development, there are a number of secondary modifications which can, start and also can start driving also the phenotype of Dravet syndrome in the chronic phase. And so there are different that have been already reported. So beside interneuron dysfunction, they've reported also alteration in other kind of circuit, so hyperexcitability of the same excitatory neuron in this case, but also metabolic changes, gene expression alteration, and proteomic alteration. And so when we are aware of these, so we wonder that, so we in the chronic phase, we can imagine that Dravet brains are actually very different from an healthy brain. And so will be sufficient. So the question that we had a few years ago is, will be sufficient with the gene therapy, so just restoring the correct gene dosage of SN1A, so to come back to an healthy condition. And so, and if yes, is there a critical time window for gene therapy delivery? And so, relevant question that started to, that we heard also yesterday. And so to answer this question, given that all the gene therapies have always limitation, we decided to use a genetic model. And so we decided to establish a reversible mouse model of Dravet syndrome. So these mice carry a wild-type allele of SN1A, while the other one carries a stop cassette between two exons of the gene. And this stop cassette interrupts, mimics a truncating mutation, so interrupts the gene expression of this allele, but it is fluxed. So there are these two LOXP sequence that can be recognized by the Cre recombinase, so a genetic tool that we can introduce into the cells and into the mice, and they can remove, and this tool can remove the stop cassette, and so restoring a completely functional allele. And so you can imagine that at different time points during the disease development, we can reactivate the gene in a very, special manner, because it's very different from a gene therapy. Here, we can reactivate the gene exactly from its locus. So we are not altering the cells in which this gene will be expressed. We are not altering the dosage of the gene. So it will be expressed exactly in a way that is dependent from its regulatory sequence, which are in its locus. And so how we could reactivate the gene? So we decided to use an adeno-associated viral vector that we heard also yesterday about, expressing the Cre recombinase under an ubiquitous promoter, because we didn't want to bias the expression of the gene. And so this virus have a property that can be systemically delivered into the mice and can really reach very well the brain. And you can see here, in particular, this PHPV variant, which outperforms the previous version. And so in this case, you can see a GFP protein, a reported protein delivered into the brain. It makes the brain all green. And so with this tool, we actually decided to perform three important experiments. So one reactivating the gene perinatally, one, and I will not show the data here, but one at P30, so in already symptomatic mice, because I forgot to tell that in Dravet mice, symptoms start at P20, and then also in, pretty symptomatic mice, so at P90, so after 3 months of disease ongoing. And so this is a Western blot that we perform in different brain compartments in the cerebral cortex, the hippocampus, the cerebellum, and striatum, to show that we were able, that first of all, our mouse model was working good. So actually the Dravet mice that you can see here in the middle have half of the protein compared to the wild type. And when we introduce the Cree virus, and so reactivating the gene directly from its locus, we could have again restoration of now 1.1 protein in all these different compartments. So indicating first that the virus was spreading very well in the brain, and also that our Cree-lock system was working properly. And so we were really reactivating the protein exactly, possibly at the gene dosage that we expected. And so with this tool set, we could start the first functional experiment. And so, as I was telling you before, symptoms start between postnatal day 18 and postnatal day 25. And here, Dravet mice start to die very dramatically. So very few mice survive this time window. And so at P30, so in mice symptomatic, we decided to deliver this Cree recombinase. And then, so we expected that at molecular level, we got this increase into NAV1.1 protein. And so that from 50% went back to 100%. And so this is the effect that we observed, first of all, on the survival monitor. So our control mice, Dravet mice, continue to die also after the critical period, which is the one that I indicated here before. So after P30, they continue to die, while all the mice in which the NAV1.1 has been re-expressed at physiological level did not. They completely recovered their pseudophenotype. We also decide to perform, to assess the effect on the seizure phenotype, the spontaneous seizure, and we have in the lab the possibility to perform a video EKG recording, so we can implant the mice with the small transmitters into the cerebral cortex and put the transmitter directly under the skin of the mouse, and you can see here a radiography of the mouse, and then we can monitor both with the camera, we can monitor the behavior of these mice, and with the transmitter, we can instead have a readout of the brain activity. And so this graph represents all the mice that we recorded for 2 weeks of recording, and each number here is each mouse. And so you can see here that every red line is a tonic-clonic seizure that the mice developed during the 2-week observation. And you can see that all the control mice had a tonic-clonic seizure, except number 6 here. Is like a seizure diary. But instead, in the mice in which we reactivated the NAV1.1 by Cree delivery, we didn't have any spontaneous seizure during the 2 weeks of observation. And this is essentially the quantification. There was a complete rescue in the spontaneous seizure phenotype. And also when we challenged the mice, so Dravet mice are sensitive to a febrile seizure. And so we can mimic this by increasing the body temperature of the mouse while we control the temperature of the mouse with a probe. And so we can gradually increase heating the mouse with a red lamp, and you can see here that all the control mice developed a tonic-clonic seizure at different temperature between 39 and 42.5 degrees, while the mice in which NAV1.1 has been reactivated did not develop a tonic-clonic seizure. So they all were seizure-free when we subjected them to these stimuli. And it's interesting to notice that we tested also these mice for some behavioral studies that we are able to perform also in collaboration with the behavioral facility at San Raffaele Scientific Institute. And so we test these mice for hyperactive behavior, for social skills and cognitive ability. And when in mice that have been reactivated at P30, we saw an important amelioration both in hyperactivity and amelioration, although not a complete rescue in the social skill ability, so in their ability to interact with another mouse, and also in their cognitive abilities. And this was measured in this time by performing this complex test, which is an 8-arm radial maze. And so the mouse have to be able to look for a food pellet at the end of these arms in this maze, and he makes a mistake every time that he re-enters an arm that already re-entered, or when he enters, he does not hit the food pellet. And you can see here that while Dravet mice, although learning, because over the days in which this test is performed, they learn because they make less error going on over the days. They actually are slower, so they make more errors, and they are slower in learning compared to wild-type mice. And this is instead, the blue curve is the learning curve of the mice in which NAMAP1 has been reactivated. And so what we learned here from this mouse model is that essentially, if we reactivate the gene also after symptom onset, so in P30 mice, we can rescue survival, we can rescue seizures, and also ameliorate dramatically behavior alteration. I don't have time to show to you now that we performed the seizure experiment also in very old mice, so in P90, and we show that there was also in this case a complete rescue of the seizures. And so this has some indication for patients, indicating that if an ideal gene therapy, so a very efficient gene therapy, administered either at diagnosis and so after symptom onset, can improve the outcome for both seizure and behavior alteration. And so a significant amelioration in epileptic phenotype can be achieved also in adult patients. And then we asked another question. So as one of the treatments which is now available for Dravet syndrome, which is actually in clinical phase 3, is an antisense oligonucleotide enhancing the expression of the wild-type allele, so belonging to the second class of the treatment that I showed you before. We wondered, and so ASOs, as you may know, require multiple dosing. So they are unstable molecules, and so they require to be administered every 2-3 months into patients. And it's pretty a pretty invasive procedure with intrathecal delivery under a total anesthesia, in particular for very severe patients like Dravet. And so we wonder if it will be sufficient to treat patients for a specific time window during the development, and so ensuring a physiological level on NAV1 during this critical time window, or if gene therapy, and in particular in this case I meant ASOS therapy treatment, should be continued after that. And so to answer this question, we took advantage of another model which was already established, which is instead a conditional inactivating mouse model, so very similar to the one that Manuela showed you before for UGDH. So in this case, we have an exon which is between a two LOXP sequence, and so upon CRE-mediated recombination, in this case, we can remove the exon, and so inactivate essentially the allele. And this CRE inactivation can also be induced anytime during development if you use a molecule, a CRE that is inducible by a molecule, which is a tamoxifen. And so we can inject the tamoxifen into mice at different time point. In this case, inactivate the gene, so passing by to 100% of gene expression to 50% of gene expression at different time point. And so we can kind of protect the mice, granting physiological NAV1 expression up to P2, up to P30 or P60, and then inactivating the gene and see what is the effect on the phenotype. And so what is clear here, these are again raster plots or seizure diary, is that as soon as the cells sense NAV1 haploinsufficiency, the mice start to seize. So also if we protect patients or mice in this case with a physiological level of NAV1 up to postnatal day 30 or postnatal day 60, actually they start to have seizure, tonic-clonic seizure, as soon as SCN1A haploinsufficiency start. And this is also clear when we challenge these mice with thermal induction. So when we increase the body temperature, again, all the three groups display the seizures at a very similar temperature, indicating that there is no difference between the three groups. And also the student phenotype start to happen also when the mice start to sense SCN1A insufficiency. And so you can see that P30 mice when we inactivate now upon 1 at P30, around P40, P35, they start to die. And this also happens when we inactivate the gene at P60. So the mice start to die. And you can see that we merge these three conditions. There is actually no difference between them. The only one beneficial effect that we found when we protect the brain development up to P30, and in particular up to P60, is an effect on the cognitive development. And you can see here in this other cognitive test, which is the Morris water maze, in which the mice have to find a platform that is hidden into a pool under the surface of the pool. And there is an acquisition phase in which they have to learn how to reach the platform, and then a reversal phase in which we make a rotation of the platform, and they have to understand that this happened. And so exploit the strategy that they have used before to learn, to relearn this process, and so to find the platform again. And so you can see that the P2 mice, which are actually those inactivated at birth, so they are the classic Dravet phenotype, are very slow in learning this process in both the acquisition and the reversal phase, while mice in which the gene is maintained expressed until P30, they actually are comparable to the Dravet mice during the acquisition, but they learn faster into the reversal, so becoming as smart as the wild type. And instead, the P60 mice are learning very fast also during the acquisition. So meaning that they're protecting the brain across the development, in particular in a specific time window with the right dosage of the channel, can actually be sufficient to ameliorate the cognitive function of the brain cells. And so we learn instead from this mouse model that exactly what I already said. So protecting the brain in a specific time window is not sufficient to prevent seizure and pseudo phenotype, but is sufficient to ameliorate the cognitive skill. And this also has an implication for therapies, because it means that lifelong SN1A gene therapy administration is necessary to provide sustained protection against seizures and behavioral problems. So with this expertise, now the question is how can we contribute to the UGDH field? Of course, making mouse models, and Manuela already introduced why the field needs a mouse model, and you are, of course, surely aware of this. So the UGDH full knockout embryo development is arrested very early during gastrulation, because we heard also today that the gene is relevant during very early phase of brain development. And so there is available this conditional activation mouse for UGDH that Manuela introduced before, and Manuela now is exploiting this mouse model to generate a brain-specific knockout, and so using a Cre line specifically expressed in neuronal lineage, and we are really curious to see if there will be a phenotype. We also know that zebrafish mutant show microcephaly, but they have no sign of epileptic phenotype. And for this reason, we started to think about the generation of the first animal model for Jamuar syndrome. And I'm really excited to hear that this mutation that we choose, the R317Q mutation, has been studied so extensively with this beautiful data from Melanie. So we chose the mutation also thanks to a suggestion from the board, the panel board, which reviewed Emanuela's grant. That is actually support. I mean, thanks to this grant, to that Telegram grant, she could support, of course, the generation of this mouse model. And this is the CRISPR strategy that we adapted to generate these. So the specific residue that we want to mutate is located in exon 8. And so we conceived a CRISPR-Cas9 strategy to essentially, we generated a single guide RNA to make a double strand cut into this specific point. And then we used a single strand oligonucleotide carrying the mutation that we wanted to insert heart as a donor. And so thanks to this double-strand break induced by the Cas9, thanks to this specific small guide RNA, we should favor the recombination of this mutation into this locus. And so we delivered both the SG RNA that we selected together this Cas9 and the single-strand oligonucleotide into mouse yes cells, mouse embryonic stem cells, which are the mouse cells from which all the embryo is essentially generated. And so we obtained possibly some cells that were edited, so carrying our mutation, and other that were not. And so we had to conceive a strategy to select the clones that carried our mutation. And so luckily enough, actually the insertion of the mutation was depleting, eliminating, disrupting a restriction site, SAU3A1. And so when we do a PCR spanning this region and then digesting with the SAU3A1, we can distinguish between a mutant allele and a wild type allele, because the mutant allele will be resistant to the cutting because we will not have anymore the SAU3A enzyme, while the wild type allele will be split in two parts, which have a different molecular weight. And so this is the results of the, a representative image of the screening. And so you can see very well that in this case, we have the digestion or complete digestion, indicating that both allele are actually wild type. In this case, we have no digestion, indicating that instead this allele may be, both allele can be mutated in this case. And instead, in this case, we have a partial digestion, indicating that one allele is wild type and another allele is instead, one allele is wild type and the other one is mutant. And this was also confirmed by sequencing analysis, and we actually identified a clear C10 clone, which was heterozygous. And so, as you can see here from this electropherogram, there are actually the peaks corresponding to both GG and AA, which is the mutation that we inserted, indicating that this clone, this mouse CS clone, was heterozygous. And so thanks to the conditional mutagenesis facility at San Raffaele Scientific Institute, we could expand this clone and then inject that clone directly into the blastocyst, which is a very, as we heard before from Melanie, a very, early stage of mouse development. And we got 4 female chimeras. So mice that were half UGDH wild type and half UGDH heterozygous. And of course, here, what we wanted to achieve was that also the germline cells of this mouse carried the mutation, because if not, they could be mutated, but they could not transmit the mutation. And so we crossed this mouse with a wild type mouse, and we waited for the first generation to address if the germline transmission was there. And so actually these mice were presenting the mutation, and so we were really happy with the result. And so we went through different crossing, back crossing in the wild type with the wild type C57 and the 6N mice in order to have a more pure background that is really relevant in genetic model in order to have no confounding the elements during the characterization. And essentially we are happy to tell today that we have got 2 weeks ago the first litters, a total of 4 litters with 32 pups from the crossing of 2 heterozygous mice, a female and a male. And while we according to Mendelian inheritance ratio, we were expecting to have on total 32 pups, 8 wild types, 16 hetero and 8 homo mice, and these are the obtained genotypes. So there are 7 wild type, 14 hetero, and 11 homozygous. So this is a good news because it's actually telling that the mutation, so homozygous mice are not lethal, so there is no embryonic or perinatal lethality of the homo mice. And they have now 2 weeks, and we are excited to start their characterization over the next months. And these are essentially the aim that we have. And so we want to first perform a biochemical and morphological characterization of these mice. So first assessment of inheritance, and then evaluating the protein level and enzymatic activity, and then evaluating the brain of these mice at both morphological level and also at extracellular matrix content composition. And then we also, in parallel, we will evaluate the phenotype, and so both the epileptic and behavior alteration. And so we will perform a longitudinal assessment of survival and growth, and also we will determine the spontaneous seizures, if any, in these mice, and also perform a behavioral test in order to determine if they present behavioral alteration. And with this, I'm finished, and I really want to thank, first of all, Emanuela Bottani, that is a dear friend and a collaborator, and that really put me into this world and involving me in this project, and the people of the lab that contributed to the data that we have up to now. So Valentina Sebastian and Elena Benvenuto, and all collaborators that actually supported the work over the years in a Dravet field, the UGDH Foundation for supporting and trusting us, and you all for the attention.